Hello, everyone. Thanks for joining us today for our ALS webinar. My name is Alex Yao. I'm a marketing strategist for ALS North America, and I'll be facilitating the webinar today. A few helpful notes before we begin. If you're having any technical issues with our webinar platform or any other questions, type your message or question using the chat function at the bottom of the screen. You can also select the hand icon to notify me of your status so I can assist you. All questions regarding the webinar material will be answered at the end of the presentation. Our presenter today is Ranil Fernando. Ranil is the General Manager for the Pharmaceutical and Personal Care Service for ALS. He has 35 plus years of experience in quality control and quality assurance testing of pharmaceuticals, medical devices, and personal care products. He oversees the operation of the microbiology and analytical chemistry laboratories at ALS. He's knowledgeable in FDA current good manufacturing practices, current good laboratory practices, and quality and stability guidelines per ICH. He's held various positions in his career, including microbiologist, stability manager, director of operations, and most recently, general manager at ALS. He holds a BS in microbiology from California State University. Today, Ranil will be discussing stability studies for pharmaceutical and personal care products. Thanks for taking the time to deliver this presentation. I'll turn it over to you now, Ranil.
Thank you, Alex, for the introduction. Appreciate that. Thank you, everyone, for joining the call. We appreciate you taking the time to join the series of webinars presented by ALS. As you know, ALS is a global leader in providing laboratory testing, inspection, certification, and verification solutions. So we pride ourselves in providing high quality, timely data and results to our clients. I do want to make a disclaimer that this presentation and the material is based on my experience and knowledge and not necessarily an official perspective from ALS Limited. So let's go ahead and get started. The goal today is to really provide a backbone of information, terms, and design of stability studies, including the types of climatic zones, the types of tests, and the setup of the specific study based on your particular formulation. I've listed some of the topics that we'll cover. I may also not necessarily cover every single slide that I have, but it will be available for your pleasure at some point in the future on our YouTube channel. One of the things I wanted to start out is to go over the information that I've used to create the presentation. So these sites provide most of the information, and I'll go over each one in the next few slides. And this is also helpful towards the end. I've listed each of the links to the various guidance documents so that as you design your own studies, you can refer back to not just the presentation, but actual guidance from each of these organizations. ICH is probably the foremost authority when it comes to setting up of stability studies, especially for pharmaceutical products. ICH covers a number of large topics, everything from quality, safety, efficacy, and under the quality section is where they have the guidance documents on how to perform and set up stability studies. It's under section Q1A, Q1F, and here I've taken a snapshot and listed the various countries that have signed on to this guidance document. And as you can see, many of the major regions and countries have participated in creating this particular document. The next organization that's very helpful is the WHO. As you may all know, WHO provides health information, health services, and different documents to the entire globe. And under that, they do have stability guidelines and recommendations. So that's another useful organization to go to. The US FDA also has its own set of guidelines. I think at this point, they're very much identical to the ICH, but again, FDA provides useful information. The European Medicines Agency also has information on stability guidelines that affect the countries that they cover. So that's another useful tool. The ISO, the International Standards Organization, also has information that will be very useful. So I've listed them as well. And then some of the bodies that cover personal care products, the Personal Care Product Council based out of US and the COLIPA Association based out of Europe also has information that's very helpful. And then lastly, the different pharmacopoeia, such as the United States Pharmacopoeia and European Pharmacopoeia and many others, also has information on how to conduct and set up stability studies. And here we start with what is the objective? In layman's terms, the point of a stability study is to determine if a product is stable, if it has the same conditions as at the time of manufacturing. In this specific definition, it's listed as, purpose of stability testing is to provide evidence on how the quality of a drug substance or drug product varies with time under the influence of a variety of environmental conditions. So temperature, humidity, light, and so forth. And it is really to establish how long is the product safe and effective for. Is it only good for a few years or less than that or longer than that? And in addition to the objective, it helps with product development. It helps to determine the properties of the product. It helps with obviously the storage condition. And if a retest duration is needed, and obviously it helps with product quality. And obviously stability studies are needed to meet various regulatory requirements. I'll start off with a few terms that'll help us in setting up these studies and designing your protocols. And I'm not going to necessarily read each one, but you can see a common method of determining shelf life is doing accelerated testing. This is to place your sample at a higher temperature, maybe at a different humidity condition as well, with the point being to increase the aging process of the sample. With the higher temperature, the molecules are going to interact and at a higher rate and possibly degrade faster than at the normal storage condition, whether that's at room temperature or whether that's at refrigeration. Another concept, which we'll get into a little bit later, is doing bracketing studies. This is where instead of testing every single batch in a particular setup, you might set up a bracketing design where only some of the batches are tested. And then there are definitions of the climatic zones, which I'll get into again in another slide, but the region is broken up into 5 different climatic zones. Another definition is the commitment batch. This is usually the batches that are created during drug product registration. You may not necessarily have all of the data, but you make a commitment to the agency that you're going to test X many batches of product, normally at least 3, and that you're going to have that data available during either submission or during future inspections. We go on to container closure system. So this is really the sum of your product, the packaging, the interior container, any external packaging or foil or any other material that's used to keep your product safe and distribution to the various clients. A dosage form, usually in a case of a pharmaceutical, it could be your tablet, the capsule, a solution or a cream. And the type of studies and the type of tests you might do will depend on the type of dosage form. Obviously, a drug product. This is the final dosage form in a final immediate packaging intended for marketing. And then the drug substance, obviously, is the active pharmaceutical ingredient that is formulated into a drug product. And then an excipient, obviously, is anything other than the drug substance in the final dosage form. It could be cellulose material, it could be saline, it could be water, or some other material that's used to create the final form. Expiration date, as it indicates, it's really the date in which the drug product or even a personal care product is still considered safe and effective. And if it's normally beyond that, there might be some issue with the safety and efficacy of the product, and it's not to be utilized. An important concept in testing is the intermediate condition. In many cases, the accelerated condition may not be stable, or the product may degrade faster at an accelerated condition. Doesn't necessarily mean that the product is not stable, but the heat at which it's stored may not be appropriate for that particular formulation. So in many cases, an intermediate testing condition is selected at the beginning. Samples are stored at that condition and generally only tested if there's an issue at the accelerated condition. Long-term testing, this is a requirement, obviously, for doing drug studies and in many cases personal care studies as well. And this is the normal storage condition of the product, and you would keep this for the normal duration of the expiration. So if the goal is to have a 2-year expiration, then you would keep long-term samples up to that point and test as needed. The shelf life, and many times shelf life and stability are used interchangeably. And in this definition, the time period during which a drug product is expected to remain within the approved shelf life specification, provided that it is stored under the conditions defined in the container label. And here we have a definition of stability by USP. Stability is defined as the extent to which product retains within specified limits and throughout its period of storage and use, example shelf life, the same properties and characteristics that it possessed at the time of manufacture. And the more important part of this is that it's dependent on specification. So part of creating a stability study and protocol is figuring out which tests are needed and creating the specs for those tests so that you can then monitor how it behaves throughout time. And here we get to that. It's usually a list of tests that are most important for your particular product. It could be analytical, it could be microbiological or biological. And these tests are repeated over time to determine if the product is remaining the same or it's changing. There's usually 2 sets of specs. You may have a release spec, which is used during the time of the batch and manufacturing and verified that the product is ready for release. But you may have a set of specs for shelf life. It could be the same list of tests, maybe a few more than normal for shelf life, but sometimes the specs itself might be tighter to get a better idea of how stable the product is. And in terms of stability, you want to consider different attributes. So for example, you want to look at chemical stability. Is the drug product, the substance within it, the excipients, are they stable over time? Any kind of physical changes, so color appearance, melting point, dissolution, viscosity, are those parameters remaining the same throughout time? You would be concerned with microbiological stability as well. Is the product remaining the same in terms of microbial effectiveness? Is the sterility, the endotoxin, any kind of microbial plating results either absent or the same as expected? And 2 other important facts are, is the product therapeutic throughout its duration? If it's a 2-year or 3-year shelf life, it's not common to do clinicals, obviously, on aged products, but we have had clients that have done some studies to prove that a 2 or 3-year product still has the same therapeutic effect. And in addition to that, then you would be concerned with toxicological safety. Are there any degradants that are occurring that's going to be above thresholds and could then be a safety issue of the product? And when should studies be conducted? Really, they should really be conducted at all phases of product development. It's routine to do certain amount of stability studies, especially accelerated studies, early stage during research and development. This is really going to help you, fine-tune your formula and make sure that the formula is stable. Same thing at pre-formulation batches, and the same as with scale-up batches, either pilot or lab batches. You really want to do all of this ahead of time rather than wait for your registration, or you're ready to launch a personal care product and you're now only doing stability and find out that there are some issues. Obviously, during registration, you're required to do so and do both usually accelerated and long-term testing. And the normal process is at least 3 batches are put in and then tested at various time points. And there's also a requirement for doing ongoing stability. Normally, it is at least 1 batch annually for each dosage and package size. And then there could be some follow-up studies as well, especially if there are some changes to the manufacturing facility or manufacturing process, or maybe some changes in the raw materials that are used for making the product. And depending on the type of product, you would obviously design your study to match that. So it could be a solid dosage form, a liquid dosage form, semi-solids, creams, ointments, lyophilized, as well as combination products that are both a device and a drug. I'm not going to really spend too much time on that last option. The focus will be on the first 4. Next, we have to really consider the climatic zones where the product will be sold, especially if it's to be at room temperature storage. So the globe is broken up into these 5 zones: zone 1, 2, 3, and then 4A and 4B. As you can see, this is based on the mean kinetic temperature in each of the regions of the world. And so if you're in zone 1, recommendation is to do studies at 21 at 45% relative humidity. If it's a subtropical Mediterranean climate, then 25 degrees C, 60%, and hot and dry at 30 at 35% relative humidity, hot and humid 30 at 65%, and then hot and very humid at 30 and 75%. And I've included a couple of examples from the WHO guideline. So they have a guideline which has each country and appropriate stability conditions or recommended stability conditions. You can see in alphabetical order some options here. They tend to be mostly in the 25, 60 or 30, 65 range. And the next slide also has some other common countries, so Brazil, Canada, China, Japan, and so forth. So you can see most studies are in the 25 degrees, 60%, or 30 degrees, 65% for long-term storage. And I've given some options in terms of setting up the study for both long-term, intermediate, and accelerated conditions. So this is a product that should be stable at room temperature. So your long-term, most likely you would set it up at 25 at 60%. But again, depending on your region, you could have those long-term samples at 30 at 65 or 30 at 75. Your intermediate is going to be a little higher at 30 at 65%. And your accelerated condition will be at 40 degrees at 75%. Generally, if you're submitting data to FDA, they like at least 6 months of stability data at each of these conditions. Occasionally, if you have the time, you would have up to 12 months of data as well. Here's an example for a product that's really only stable at a refrigerator. So here, your long-term is at 5 degrees plus or minus 3. But your accelerated then is at a higher temperature. Now, it could be that we'll talk about it a little bit later. It may not really be stable at 25 or 30. So you might only need to do 1 or 2 months of testing to see that it just fails. And then you would just continue your long-term study. Here's an example for a freezer. Obviously, products that are only stable under freezing conditions, you really can't really have any intermediate or accelerated conditions. You're just having to keep it at the freezing condition. If it's a semi-permeable container and you want it stored at room temperature, then the conditions are slightly different. You have a lower humidity requirement for these type of products. The temperatures are really the same. And in terms of the frequency, the guideline is that during the first year, it should be that every 3 months the product should be tested, then every 6 months over the second year, and annually thereafter. So for a product that's going to be stored at long term, you would do your baseline 3, 6, 9, 12 for the first year, 18, 24, and then subsequently annually, so 36, 48, and so forth. Your intermediate and accelerated, you would pretty much do those monthly for the first 6 months. Now, again, intermediate, you would normally only store those. You may not need to do any testing, but if you need to, then they're available for subsequent testing if the accelerated testing fails. And basically, for any new drug entity, you'd want to pick the appropriate chemical, physical, biological, or microbiological tests that are most appropriate for your particular product. And I'll go over some options depending on the type of product. So here's an example for an API for a drug substance. So here you set up your physical, chemical, and microbiological test scenarios. And in most cases, you're doing colored appearance, particle size if necessary, maybe an identification test with UV, IR, or some other means. You might also do assay and degradation, and also do that the assay is stability indicating by doing oxidation, photolysis, acid-base, temperature, and pH extremes. If there's any residual solvents to be concerned about, elemental impurities, and any other monograph test, as well as usually microbiological testing for aerobic plaque count, yeast and mold, and any specified pathogens. Here's an option of test for injectable drug product. Again, you're always going to do appearance, color, but here you would do container closure if it's in a obviously injectable in a closed container system, as well as appropriate chemical analysis for the assay identity and impurities. If there's any antimicrobial agents, you would want to consider testing for those. And again, any impurities, either organic or inorganic, and verify there at below the thresholds that are specified. And for injectables, for microbiology testing, you're primarily doing endotoxin, particulate matter, and sterility. If it's a lyophilized product, very similar to the one set of tests before, but here you're going to worry about reconstitution of the lyophilized powder, appearance of that solution, as well as container closure integrity and the normal set of chemical and microbiological studies. Oral solutions, here you're looking at, again, the normal colored appearance, pH, maybe taste as well, and then the normal set of chemical assays, degradations, impurities. And for micro, you would now be doing aerobic plaque count, yeast and mold, specified microorganisms. If it's a water-based product, you're doing antimicrobial effectiveness testing as well. Oral suspensions, very similar, slight differences in the physical test, things such as dispersibility and any particle size concerns, you would do those as well. If it's an inhalation type product, slight different tests on the physical, you're going to worry about the container closure as well on these kind of products. Again, if it's water-based, you would do antimicrobial effectiveness tests on the microbiology. If it's not, then you would do maybe water activity. If it's a tablet, then your physical tests would change. You're doing, in addition to colored appearance, you're doing disintegration, hardness, friability, dissolution, possibly moisture content. Your chemical and your microbiological tests are mostly the same, but under micro, you're worried about water activity, verified that the product is a dry product. If it's a hard gelatin capsule, again, the tests are slightly different. You're doing colored appearance, possibly taste, dissolution, and moisture content. Similar set of chemical assays and for impurities and any other monograph tests that are required, as well as your testing for your aerobic and yeast and mold and specified microorganisms. Very similar for soft gelatin capsules as well. If it's a topical product, your test for physical would change. So color appearance, pH, viscosity, specific gravity, solids or moisture. And if it has an active ingredient, obviously assay, identity, any degradation and impurities, and organic and elemental, both organic and inorganic impurities. And the same for microbiological as before, plate count, yeast and mold, specified microorganisms, and antimicrobial effectiveness test. One of the concepts to remember when you're doing stability studies, and in fact, actually before you do your stability studies, is to do method validation. ICH-Q2 and ICH-Q3A, Q3D cover this topic, but also FDA and other agencies have information on doing analytical method validation and proving that the method is valid for that specific formula, and it's also stability indicating. And it's key to do this on each individual formula. There is no standard method that's validated by a lab that's going to be good for any product that just comes through. So it's really important to work with your laboratory, make sure that the method is truly validated for your formula. The same applies for any microbiological studies. So your sterility, endotoxin, preservative effectiveness test, your microbial limits test, they all need a suitability procedure, again, per each formula. So you want to make sure that that's conducted. Another concept with stability studies is actually photostability. You want to make sure that your product is photostable. Usually this is done at the formulation stage. The formulators usually know the basic attributes of the specific drug compound and whether it's photostable or not. But it's important to perform that study at the beginning to prove that it is in fact stable. But if it's not, then the appropriate actions are taken, such as packaging the product in a amber vial or containers that are not permeable to light. Normally, ICH requires exposure of samples to both ultraviolet light as well as visible light. Here are some of the specs on that. So it's important that you have the product usually covered in foil, exposed to the light, as well as without the foil exposed. And then you do the appropriate analytical testing to prove that the active ingredient doesn't change, it's at the same level, and there isn't any degradants. I'm not going to go into the details of how to do some of the measurements here, but it's available for you. There is a flow chart by ICH on the process for verifying if a product is photostable or not, and I've included that here. And it helps you as you do your initial studies to determine, if it's truly stable or not, and if you had to make any changes to your packaging. Another part of obviously setting up a product and doing stability studies is the container closure system that's used. So a system that is meant to be intact, you would want to verify that it is truly free of leaks. So you have a number of tests that can be performed, everything from dye immersion to microbial leak test to helium detection by analytical means. So you want to usually consider these as another test as you're setting up the different types of products. And then if a product, we talked a little bit about it, if it's impermeable or permeable to moisture, you want to make sure you set it up on the right humidity conditions and you verify weight loss or weight change as well. We'll talk a little bit about how to set up your study design, and that's what I'm going to do in the next few slides. So here's an example of some information that's very helpful when you provide your product to a laboratory. So here we have the product name, the strength, the batch number, and the batch size. But it's very helpful sometimes if you put all of this other ancillary information like the manufacturer of the product, the API lot number, API manufacturer. If all of this is provided, we can put that on your report so you have it available for the future. Many times we get study projects, but we only get the name of the product. We only get maybe a batch number. So the more you provide, the better it is for us to add that to your protocol and have it available for the future. And then here's an example of a drug product specification. So you have the list of tests on the first column, the appropriate method on the second column, and then the appropriate specification on the third column. And as I mentioned, these specs could be different for release versus stability, but you'd want to have this so that as you're doing the study, you know if something is failing, you can then trigger an OS investigation or an out-of-trend investigation and take appropriate action. Here's kind of a study design layout. As you can see, we've taken that set of tests and then listed the time intervals. Here it's at 25, 65% humidity. And you can see most of the physical and analytical tend to be performed at each time point, where some of the microbiological, you may not need to do that at each time point. So here we have it at beginning, middle, end, or some time points in between. This obviously helps reduce cost as well, not having to perform every test at every time point. And then a part of that is calculating the sample need. So here, taken that design layout and added the quantity of samples needed depending on the specific study, and then you have a total at the end. We've exaggerated the extras here a little bit, but it's really important to remember to have some extra units available at the beginning. So maybe 10 or 20% extra if possible. This will really help you in case you had to either do some additional retest or if you want to extend the duration of the study. We've had cases where we are at the 24-month time point, everything's great, everything's passing. Client comes back and says, yes, I want to make it 36 months now. Well, unfortunately, there isn't any sufficient number of products to perform all the tests. And you're back to the drawing board. Now you're going to take 3 more years to get three years of data because we don't have any more samples. So it is something to consider when you're setting up your study. Here's an example of a bracketing design, which I briefly talked about earlier. So here's an example where we have a product. It has two different concentrations or strengths. And normally you would do 3 batches of the first strength and 3 batches of the second strength. And you'd have 6 batches of product that you would need to test at each time point. But under bracketing design, you're not necessarily doing that. You're doing all the tests at the beginning, all the tests at the end, but in between, you're alternating doing some batches at, let's say, 3 months and not others, and then the other batches at the 6 months. And this, again, helps to get basically good data, but not necessarily have to have every single sample, every single batch tested for each time point. And obviously helps reduce costs. Here's a matrix design. So in this particular case, what we have is not just the changes in strength, but changes in container. So you can see up on top, you have strength 1, 2, and 3, and then you have a container type A, B, C, and so forth. And obviously you have 3 batches, and you could theoretically be testing, every single batch, every single strength, every single container type. But here you set up a matrix design where certain batches and certain strengths are tested at certain time points so that, again, you mix and match to get the best data that's available rather than have to spend time and resources, obviously money, on testing every single batch separately. I'll briefly discuss that, obviously, when you do perform stability studies, you could have stability failure. These are some examples of what could happen during a product. Many times this is known ahead of time, so you really don't get to a failure at the commitment batch stage, but it could happen during the initial R&D phase. And obviously you would work with your formulators to figure out why something is failing. And some other factors that affect product stability is obviously microbial contamination, the container, and product incompatibility, and obviously failure of the container closure system as well. ICH Q1E covers data evaluation. So, for registration batches, it is expected you have 3 batches of data, and that you use all of that information to verify that your product is stable for the expected length of time. That includes all of your chemical, microbiological, biological, or physical testing. And in terms of data presentation, most stability studies, you're going to have it in a tabular format, various test results by each temperature time point at each interval. But in some cases, it's helpful to do graphical presentation of the data and have a narrative of the data as well. And obviously, you want to evaluate all of the data points, long-term accelerated, and if there's a need, perform statistical analysis as well. And in some cases, do extrapolation of the data as well. And this is kind of a decision tree that's available in the literature that I mentioned. And again, it's too small for you to see here, but it helps you to decide how best to approach the data evaluation. Here's an example of a extrapolation. Here, this is results for a assay. You can see the data indicates they did testing from 0 up to 12 months, and then they are doing extrapolation to see at what point does it dip below the 95, 105% range that they're looking for. Same type of extrapolation, but in this case, it's impurity or degradation information. So here you have data up to 12 months, and then they're looking to see at what point does that impurity expected to be above a threshold. I'll switch a little bit right now and go over the personal care type products. Obviously, these are products that may not have an active ingredient. I mean, obviously, you have OTC products that are still considered drugs by FDA, but then the personal care would be without any active ingredients. And these are the laws that govern that. In addition, I didn't get to put on here the new MOCRA regulation as well. Most cases for personal care products, the storage condition will be 25 degrees for real time, 40 for accelerated. It's rare to have products that are kept at refrigeration or freezing when it's a personal care product. So this tends to be the most common temperature time points. What is also common is to perform freeze-thaw testing. You want to verify that as your product is shipped throughout the country, it goes out in air transport, that the freezing does not drastically or alter the product and cause it to break down. And so that's important. We normally recommend at least 3 cycles for freeze-thaw. And then another important part is doing any period after opening. So if it's a product that's going to be sold in EU, you want to do the period after opening, which normally consists of opening products at the beginning of the study and keeping those again at the appropriate storage condition, and then checking that they're still stable. I'll go over some of the basic tests you might do on a different types of personal care products. So here is a topical water-based product. You would do your physical tests such as colored appearance, pH, viscosity, solids, specific gravity, freeze-thaw. Usually there isn't a lot of analytical that needs to be done, but these days there are many, many regulations that govern impurities. I'm sure you all know that there are state regulations in California, Washington, Vermont, and other locations that ban impurities such as 1,4-dioxane, parabens, phthalates, and formaldehyde, and so forth. So these days we are performing those tests, even though the product doesn't have an active ingredient. And then your normal microbiological testing, plate count, pathogens, as well as antimicrobial effectiveness testing. If it's anhydrous products, it's a lot less involved. You're doing maybe color, appearance, some of the impurities that are of concern, especially for products that are color-based products, your foundations, things like that may have earth-based materials. You want to worry about metallic impurities, so elemental impurities for lead, mercury, cadmium, arsenic, nickel, and so forth. And if it's meant to be anhydrous, you would consider doing a water activity test to prove that it truly is anhydrous. Fragrance, again, very few tests that you would do. You might do the fragrance allergens maybe at the beginning, verify that there isn't any, allergens in the product. Similar layout in terms of how you put the protocol together. The more information that can be provided, the better off in the future. So not just product name or formula, but where it's manufactured, the type of container, and appropriate conditions for storage of the product. And the set of specifications, it'll be obviously a lot less tests in many cases, but again, it's important to have that set of tests, whatever appropriate method and whatever the specification is for those tests. Here's a brief study design. This is for a short-term accelerated or long-term study at 25C for 6 months. Again, physical tests tend to be performed at each time point. Microbiological usually beginning end or beginning, middle, and end. And again, a calculation of how many samples are needed and some extra samples if needed. And the presentation is very similar as well for pharma studies, where you would most likely have data in a tabular format, but if necessary, it can be graphed, and you would evaluate all the data, accelerated, long-term, and any things that need statistical analysis. So that's a quick summary of how to set up a stability study. I didn't necessarily cover every single topic that needs to be done. I wanted to leave a little bit of time for any questions. So again, we've utilized the ICH, WHO, ISO, and various governmental regulations to come up with how to set up the studies. I do want to thank all of you for joining. I've listed my contact information, our Director of Operations contact information, and our general contact details. If you have any questions for us, please let us know. And I'll just cover the fact that I've listed a lot of the links to the various guidance. So hopefully it'll help you when you're doing your due diligence to look up some of this information as well. And feel free to reach out to ALS if you have any questions on the various study designs. We are more than happy to help you and help you create a great protocol.
Thanks, Ranil. We're going to open up the forum for questions now. If you could please write out your questions using the Q&A function in your toolbar at the bottom of the webinar. Attendees can also see all submitted questions using this window. I'll just give everybody a few moments to type in their questions. And while they're doing that, I will review a couple of brief notes. This webinar presentation has been recorded. All participants of this webinar will receive a follow-up email once the recording is available to view. We also post our webinars on our ALS YouTube channel and the webinar playlist. I encourage you to find a wide range of video resources and past webinar presentations available there. Also, please follow our company page on LinkedIn as we post announcements and registration links to future webinars and other valuable resources and updates. So it looks like we have one question. In the simplest terms, is it 3M or 6M accelerated sufficient for OTC 2-year shelf life to launch? I've heard and seen both.
There is no easy answer for that. I have the rule of thumb tends to be that we've used sometimes is 3 months accelerated equals roughly 1 year room temperature. This is comparing 25 room temperature and 40 degrees accelerated. So if you go by that, then for doing 2 years, you would need roughly 6 months of accelerated data. We use that rule of thumb also because the Agency for Drug Products asks for 6 months of data, but we also have clients that are just very comfortable with their product. It's not a brand new product. It's a tweak of a formula. They have experience with the product. So they go and assume that if they have good data after 3 months of testing and accelerated, it's going to give them 2 years of shelf life. But I've looked at this for over 30 years. There isn't a single place that explicitly indicates that what the equation is, that 3 equals 1 or 3 equals 2 years.
Thanks. We have a question from Haley. She's asking if there's any guidance for fill volume or weight on a stability study.
Yes, so normally this would be done really more as your initial product release criteria per USP or various monograph requirements. And what we do perform is more of a weight change or weight loss study. So we would write down the initial total weight of the package, multiple packages, and use those units throughout, the same exact units throughout the duration of the study for each temperature. So if the product weighed 20 grams and 20.01 grams, 2 units, we would weigh that each time point and verify that the weight itself doesn't change. But we do have clients that want us to verify the actual content. So in those cases, what we do is we take the weight, we empty out the product, and then wait after all the product has been emptied and verify if it's meant to have 5 ounces, it is indeed 5 ounces. So if it's meant to be 20 grams or 10 grams, then we verify that. But it's usually more a test that's done during initial manufacturing rather than part of a stability.
Thanks. We have another question from an anonymous attendee. Are studies only done with 3 batches from the manufacturing plant, or can they be done with batches in R&D?
Well, its answer is both, because you should be doing some studies with R&D batches to know that the product is stable before you go to manufacturing, because you really don't want to get to a point of manufacturing and never had done any stability and only to find out it's failing. Now you not only have a stability failure, but you made X many units of product, you start packaging, labeling, and what are you going to do at that point? But theoretically, though, for OTC products and pharmaceutical products, and really even personal care products, it should be in the final packaging used for sale. So usually that's not lab batches, that's really the final manufactured batches. That's a requirement by FDA for drugs.
Okay. Another one from Haley: can 36 weeks accelerated project 3-year expiration, or does it need to be real time for 12 months and then accelerated 12 weeks?
I would say what I said earlier, which is that the rule of thumb has been 3 months equals 1 year. So you're looking at approximately 12 weeks would be. So yes, I mean, that's pretty close in terms of if you were to use that 36 would be roughly 3 years. But you can't just go beyond by just accelerated. You're meant to also have samples at room temperature while you release the product with the expected stability date or expiration date, but you still would have samples at room temperature and keep that going so that 3 years later, you know that, okay, what I expected from my accelerator data was proven because I have now real-time data. I know that personal care people are not necessarily doing that. They're just going with accelerated only. But if it's an OTC or if it's a pharmaceutical product, you really do need to have both accelerated and room temperature data.
Okay, sounds good. We have another question. Are there specific or different stability study designs for products with probiotics or fermented ingredients that may degrade or break down over time with heat?
Not that they're necessarily different, but your study conditions will be different. If a product truly is not stable at whatever that temperature is, if it's not stable at 25, for probiotics, you're going to be doing your microbiological analysis and verifying the level of each probiotic, lactobacillus, or whatever the other organism that's in it. If it's got 1 billion colonies, that is still stable and it's alive 3 months, 6 months, and so forth. But if they're not, you would need to know that, because then you're selling a product that's really not stable, and you might need to then verify if it needs to be kept at refrigeration temperature. A good example would be not necessarily a probiotic, but something like benzoyl peroxide. It's inherently unstable at higher temperature. So you really can't do accelerated testing. You'd want to do pretty much room temperature testing. It's probably even better at refrigeration if you can keep it at refrigerated.
Okay. We have a question from Saba. Are there any other types of testing recommended for personal care anhydrous products?
Not necessarily, because if it's anhydrous, chances are it's either a powder or it could be maybe an oil. So if it's more of a powder type material, then you really can't do viscosity, specific gravity, that type of physical test. You're primarily doing colored appearance. You'd want to do water activity to prove that it truly is anhydrous. So that's all you would need to do. Obviously, if it's a oil that's anhydrous, you could consider some other tests such as fat concentration, peroxide value, things like that.
Okay. What is the minimum shelf life / expiration date you can or should launch an over-the-counter product with?
In our experience, the minimum has been 1 year. You want to make a product and you want to sell that to the retailer, and they may have their own rules. They may not want to accept a product with anything less than a 1 year. I'm not sure if the larger retailers would even accept 1 year, because if it doesn't sell quickly, and 1 year will go by rapidly. Now, what do you do with that product? Do you get it retested, or do you have to dispose of that product?
Okay. And it looks like that's all the questions we have for now. I want to thank everyone for attending today. Again, you can contact Ranil for any questions. We'll provide his contact information in the follow-up email, along with a recording of today's webinar. I hope everybody enjoys the rest of their week, and thank you for attending.